All right, welcome back. Here we go. Digestive system. This says chapter 15. Well, that is not chapter 15 in our book, so we're going to ignore that because this is actually in our books, chapter 4, isn't it? Chapter 4, I believe. And chapter 4 deals with the digestive system. So let's take a look. When we think about the digestive system, we think about it as one big tube, starting with your mouth, and that tube goes all the way through the body, ending with the anus. We call this tube a couple different things. We could call it the GI tract, which means gastrointestinal tract, or we could refer to it as the alimentary canal, right? Alimentary canal. But if food is passing through this tube, it's passing through the organs of the GI tract. Now, the organs of the GI tract are not the only organs of the digestive system. There are other organisms. There are other organs that are playing a part, but playing a part from the outside, right? So food isn't technically going inside of those organs, but they are adding secretions to the food products as they are traveling by. We're talking about things like the pancreas, the liver, and the gallbladder. And because food is not going in them, we would refer to these things as being accessory organs. All right. So what's happening in the GI tract? Well, number one, we're putting the food in there in the mouth. All right. Number two, we're kind of mixing them around. Number three, we're breaking them down. Number four, which is I think is the most important part, is we're getting the nutrients from the GI tract and putting them into the blood, right? Because it's the nutrients that we need to deliver to all of our cells, right? So that's the most important thing. Lastly, whatever's left over, we get rid of, and of course we would refer to that as defecation. All right, so this is a pretty good picture here. We're showing you the organs of the alimentary canal in black, the mouth, the pharynx, which is kind of like the throat, the esophagus, the stomach, and then through the small intestines, through the large intestines. The rectum would be over here. And then lastly, out through the anus. Now we see also accessory organs in red. So the teeth and tongue, the salivary glands play an important part. There's 3 salivary glands, parotid glands, submandibular glands, sublingual glands. They're adding secretions, breaking down the food very early on. The liver and gallbladder producing and storing bile, the pancreas contributing enzymes that break down many different types of molecules that are in the food. All right, so this is a great picture. So when we look at the structure of the GI tract, if you were to take somebody's intestines in your hand, cut them and look down into them, and let's say you stuck your finger on the inside, that would be kind of gross if you did that. But if you did do it, you would notice that the inside, the innermost layer, is filled with mucus. It's very gooey in there. For that reason, we call that layer the mucosal layer. That's the mucosa. The layer directly behind that is called the submucosa. And that's where we're going to find things like glands, nerves, and blood vessels. Behind that, we have the muscularis, and that's because we have muscles back there, right? So remember, you can't control the movement of food through your intestines. So it's a very special type of muscle tissue that we find. It's called smooth muscle. Remember, your bones you can control because the type of muscle that we see attached to our bones is called skeletal muscle. So we have the ability to control those muscles. These muscles we don't. They're completely involuntary. In fact, they are under control from your brain, but not the brain that you have control over. It's more of like the brain stem. All right, so this is smooth muscle tissue in here. It's surprised that doesn't say that. I'm going to write that in there. Smooth muscle. And lastly, the outermost layer of the GI tract, we would call that the serosa. Let's take a look at this. Yeah, here we go. Here's a nice cross section of the small intestines, and the layers are kind of separated as the inner layer is pulled out. So this is where food would be traveling inside the lumen. Anytime we're talking about an opening of a tube, we refer to that as a lumen. So this is the lumen here. And this represents the mucosal layer. All of this. Now, behind the mucosal layer, this is where we're going to find lymph nodes, blood vessels, nerves, arteries and veins, that kind of stuff. That's our submucosa. And then we have these layers and layers of smooth muscle making up the muscularis. And then we have this outer tissue called the serosa that kind of holds everything together. Peristalsis is a process. I guess we could describe it as this is how food is pushed through the GI tract. Okay, so imagine you have a garden hose and you want to send a marble through into one end of the garden hose and you want it to come out the other end. But what happens is as you send the marble through, you notice that the marble is just a little bit too big and it gets stuck, but you still want it to come out. So what you would have to do is use your fingers to push behind the marble to get it to continue to move forward until it comes out the other end. So this is how we kind of move our food through our digestive system. Here we see a picture of the esophagus. There's muscles making up the walls of the esophagus, and the muscles will contract behind the food, putting pressure behind the food, and that's what's going to cause the food here to be sent in one direction, because this wave-like muscle contractions called peristalsis that pushes the food through the entire system. So they're showing this in the GI tract, but this will continue in the stomach and then continue in the small intestines and the large intestines as well. Mechanical digestion is one type of digestion, right? Mechanical digestion is the physical breakdown of food. Most of the mechanical digestion that's happening in our bodies is happening in the mouth because of the teeth, right? We are taking in the food and then we are physically breaking it apart. When we chew the food, what are we doing? We're increasing the surface area, right? We're taking a ball of food and chewing it up into many, many different pieces, increasing the surface area so the enzymes of the digestive system can work on that more efficiently. All right. So we have incisors in the front, and then we have some canines, right? Those are the Dracula ones, the pointy ones. Canines, incisors. Oh, canines over here, incisors. And those are for cutting, more for like meat eating, cutting into meat, tearing into meat. And then we have these grinding teeth back here, which we use for we call those molars and premolars. If you're eating plant material, you would grind it up using those teeth. The tongue is an important part of digestion because the tongue manipulates the food, right? So once food is in your mouth and it's mixing with saliva, we refer to that as bolus. Bolus is another word for food. All right. So the bolus gets positioned throughout the mouth. The tongue helps it to mix with the saliva. The tongue helps it to position over the teeth that you want. There's also on the surface of the tongue specialized cells called keratinized cells. Keratinized epithelium. And what that does is it provides a rough texture, and that helps to just move the food around your mouth because the food becomes very slippery as it mixes with the saliva. All right. There's also an enzyme produced by the tongue called lipase. Remember, if a word ends with ase, it usually means it's an enzyme and usually means that it's going to be working on something. A lot of times when we look at the names of enzymes, the beginning of the word, in this case, lip, that stands for lipids, right? So this enzyme is breaking down lipids. Lipids are fats. All right, so lingual lipase is an enzyme coming out of the tongue. And what it's going to do is it's going to start breaking down triglycerides. So triglycerides are fats or oils. In the back of the oral cavity, we have our first line of defense against any undesirables that might be in the food, and this is called the tonsils. All right, so it's inside the tonsils that we have specialized lymphatic cells like macrophages and lymphocytes that are looking for things who want to invade our gut. All right, so and they will greet those things. If there are bacteria in the food, that'll activate those cells in the tonsils, and the tonsils will swell as they attempt to destroy the bacteria. MALT. MALT stands for mucosa-associated lymphatic tissue. So starting with the tonsils, we see MALT. Then it continues throughout most of the GI tract. We can kind of break down MALT into a couple different subcategories. GALT is gut-associated lymphatic tissue. So throughout the whole gut, if there's bacteria that are trying to invade the gut, well, there's lymphatic tissue that are looking for that kind of bacteria. There's also BALT, which is bronchus-associated lymphatic tissue. So that's more of a respiratory lymphatic tissue. If there's bacteria that were in the air that you breathe in that are trying to penetrate your lungs and invade your lungs, BALT would greet those. There's also NALT, which is nasopharynx-associated lymphatic tissue. So that's more of the upper respiratory tract. So we do have these teams of lymphatic tissue, teams of white blood cells. Remember, these are cells in the immune system that are designed to kind of hang out in these specific areas, look for any invaders, and when they do see the invaders, they attack those invaders. And so the MALT, these immune cells, these clusters of immune cells, or these nodules of lymphatic tissue, are very important for preserving homeostasis, right? Because when we have disease, when we have infection, that is a disruption of homeostasis, right? Disease is a disruption of homeostasis. So MALT tissues represent a very large percentage of the entire immune system. And when we look inside the MALT, we're seeing about half of the body's lymphocytes. Lymphocytes are like B cells and T cells, and half of the body's macrophages exist in this form called MALT. Macrophages, with macrophage, macro means the opposite of micro. So this means big. And phage means to eat. So these cells are literally called big eaters. And what they do is they're able to leave the bloodstream, and they're able to leave the lymphatic vessels, the lymph, and actually patrol tissues. And when they do find something that they don't like, a pathogen that doesn't belong, they'll engulf it. All right. So these are very important types of immune cells. All right. The salivary glands, that's one of our first accessory organs. They secrete watery saliva into the oral cavity. When you see food, when you smell food, you start to increase the production of saliva. And what that does is that mixes with the food and creates a slippery bolus. Remember, bolus is just another type of word that describes food after it's entered the digestive system. All right. So this is food as it's mixed with saliva. All right. On the sides of the mouth, we have parotid glands. And then under the jaw, we have submandibular glands. And then we also have sublingual glands under the tongue. All right. So what is it about saliva? Well, besides making the food slippery, which is nice because we need the food to slide through the GI tract, there's also a lysozyme. Lysozyme. Lysozymes are enzymes that help to destroy bacteria. So again, this is part of the first line of defense. When we put something in our mouth, if there's bacteria in that, the saliva could be one of the first things to help protect us from that bacteria, along with the tonsils. There's also an enzyme amylase. Remember what I told you, if it ends with ase, it's usually an enzyme. And the first part tells us what the enzyme is working on. Well, there is a carbohydrate called amylose. Amylose is a polysaccharide. So if you're eating pasta or something like that, the starch in it, which would be something similar to amylose, then that amylose is going to start to break down. So immediately in your mouth, as food is mixing with the saliva, it's beginning to break down, specifically carbohydrates. If you put a piece of bread in your mouth, don't chew it, just let it sit there. You could probably feel it chemically digesting as the amylase in your saliva is working on it. You could feel it getting smaller. All right, so the next stage would be swallowing, right? So once we swallow, we give up all voluntary control. You have the ability to control the muscles for swallowing, but once it enters into the esophagus, it's all under the control of the GI tract. So you give up voluntary control, and then it's the rest of that movement is controlled by the autonomic nervous system. But then when the food is ready to exit, it exits through the anus. Of course, we have that's part skeletal muscle, so we do have control of that. And thank God for that, right? All right, so we could break down swallowing into 3 stages. There's a voluntary stage, so you are voluntarily moving the food around, and then you voluntarily swallow it. Well, where does it go? Well, after you swallow it, enters into the pharynx. The pharynx is a common passageway for food and air, right? So it's really like the back of your throat. There's 2 pipes at the end of the pharynx, however. One of them is the esophagus. That's where we want the food to go. And one of them is the trachea, which is for air only. We do not want food to enter into the trachea. If it does, that's how we choke. All right. So this is not unusual. We've all choked on something at some point in our life. But what prevents the food from going into the trachea is this little flap of tissue called the epiglottis. If I were to look at the trachea, I'm choking right now. The trachea is the windpipe that brings air from the larynx down to the lungs, right? So, and again, this has some cartilage rings in it. The opening at the very top over here, the opening here, this is the trachea I'm drawing here. The opening is called the glottis. So this flap of tissue I'm talking about that covers the glottis when we swallow, preventing food from going down there. The food would pass over here and then bounce off the epiglottis and go into the esophagus. This is called the epiglottis. Epiglottis meaning above the glottis. All right. Then the food would enter the esophagus. From the esophagus, it would go down into the stomach. And how do we get it from the esophagus into the stomach? Well, that peristalsis. And then as it is about to enter into the stomach, it passes through a circular muscle. Whenever we see this word sphincter, any sphincter is a circular muscle. Oh, what am I doing? Circular muscle. So as the food comes down, at the base of the esophagus is the lower esophageal sphincter muscle that will relax, allowing the bolus to enter into the stomach. And after the food drains into the stomach, then the bolus will contract, and that prevents the food from coming back from the stomach back up into the esophagus. If we have problems with our lower esophageal sphincter muscle, sometimes when we're laying down, for example, the food that is mixed with the acids of the stomach will want to creep back up past that lower esophageal sphincter muscle and drain back into the esophagus, and that burns, right? When that happens, it burns and it's very uncomfortable for us because the acids should not be going up there. That esophagus doesn't have protection against the strong acids of the stomach. And that's what heartburn is. All right, so here we see the bolus and we see swallowing happening. Here, when you're breathing and talking, the trachea is open, right? The epiglottis is open when you're breathing and talking. But when you swallow, the epiglottis covers the opening of the trachea, and that causes the food to continue down the esophagus, which is where we want it to go. When we look at the stomach, there's 3 layers of the stomach, and the layers of the stomach are made out of muscle, and these muscles are arranged in 3 different directions. They refer to that as a circular arrangement, a longitudinal arrangement, and an oblique, right? So oblique, usually that means it's on like a slant, all right? Why is that important? Because when we have muscles that are arranged in 3 different directions, it allows the stomach to twist, right? And what that does is it churns the food that is inside there. If we only had 1 layer of muscle, all it could do is squeeze in one direction. All it could do is squeeze, right? But if we have 3 different layers of muscles going in 3 different directions, it causes this twisting motion. So on one end of the stomach, here we have the esophagus coming down. Here's that lower esophageal sphincter muscle. And then we have the large J-shaped body of the stomach. And then at the end of the stomach, we're going to have another sphincter muscle. This is called the pyloric sphincter muscle. And this is what's going to keep the food inside the stomach here. So food goes through the lower esophageal muscle first, and then this is contracted, and that's what keeps the food in there for the appropriate amount of time. This muscle is so powerful that if the body detects any undesirables in the food and we want to vomit, we want to get that food out of our system, it's this muscle that is responsible for projectile vomiting. Here we see the longitudinal, circular, and oblique layers of muscle. If we look inside, we see a folded inner lining. The folds are called the rugae. And what the rugae do is they allow this organ to expand, right? The stomach expands as it fills. So the churning and mixing, when we've churned food in the stomach, this is another example of mechanical digestion. Because what this is doing is it's physically breaking the food apart through the action of these muscles. But there's also enzymes in here that will begin to digest proteins. Remember, in the mouth, we had enzymes that are breaking down carbohydrates. Now that we're in the stomach, we could start to break down proteins. If we look inside the stomach, there is a strong acid called hydrochloric acid. And this thing does a couple different things. Well, it breaks down large molecules. It destroys microbes. So if there's anything in the food that doesn't belong, any bacteria, the stomach could probably destroy that bacteria just with the acids that are in there. However, this stomach acid is so strong that it could possibly harm the lining of our own stomachs. So in order to prevent that, we have to create a thick wall of mucus on the walls of our stomach to separate the wall of the stomach from our own hydrochloric acid. There's also if we look at all the juices of the stomach, we would call this is all gastric juice, right? gastric juice. And what is gastric juice? What is it? Well, one thing is what there's water in there, right? Because remember, water is very important for all types of reactions, especially these hydrolysis reactions or digestive reactions. There's an enzyme that breaks down proteins, which is called pepsin. The thing about pepsin, um, even though it breaks down proteins, it doesn't work unless it's in a low pH. So we provide it with a low pH. We provide it with hydrochloric acid. Without this acid in the stomach, this pepsin wouldn't work, right? And then, well, if we have acid, a strong acid like hydrochloric acid, we have to protect the walls of the stomach. So there's also mucus. So these four things kind of make up gastric juice. This is the enzyme that's breaking down proteins, right? And this is an additive that we provide in order to activate this enzyme. So where's the pepsin coming from? Well, if we look in these at this microscopic image of the stomach, we could kind of find two different cells. There's chief cells, and there's parietal cells. The chief cells produce the pepsin or pepsinogen, which is the precursor of pepsin. All right, so it's these cells that are producing the enzymes that are breaking down proteins, and they're also producing an enzyme that breaks down lipids. It's parietal cells, which is a second type of cell that's found in the gastric pits, that are producing the hydrochloric acid. All right. So remember, the hydrochloric acid is important because it activates the pepsin. Without a low pH, this pepsinogen or pepsin, is not going to function. But what's good about the low pH of the stomach is that it kills any microbes that might be down there. These parietal cells also produce this stuff called intrinsic factor, and intrinsic factor is very important because we need it in order to efficiently absorb vitamin B12, which we need vitamin B12 to produce red blood cells. All right, so what's happening as the bolus reaches the stomach? Of course, the walls of the stomach are mixing and churning, and we have waves that are occurring every 15 seconds or so. We have food that can stay in the stomach for as long as an hour before they move into the second part of the stomach, which is the body. Typically, the food stays in the stomach between 4 and 6 hours before the food leaves the stomach through the pyloric sphincter and enters into the small intestines. So what's happening? Well, this is a negative feedback loop, right? And the first part of digestion is called the cephalic phase. So if we see food, if we smell food, if food is entering into our mouth, the first part of digestion called the cephalic. Remember, cephalic means head, right? Or in this case, the brain part. And what's going to happen? Well, there's going to be hormones that are sent from the brain because of this food that's entering that are going to go down and increase gastric secretion. So it's going to cause the parietal cells and the chief cells to increase their secretions because food is going to be on its way down. So the second phase would be the gastric phase, and this is when the bolus reaches the stomach. As the food is in the stomach continues to produce this hormone gastrin, which is going to continue to increase the stomach's secretions. Now, after a couple hours, the food will leave the stomach, and then we enter into the intestinal phase. It's this final phase that, as food enters, passes through the pyloric sphincter muscle here and enters, that a different set of hormones is going to decrease the gastric secretion. So the activity of the stomach will slow down. Okay. And as feedback is coming up from the stomach, as it empties to the brain, a new set of hormones will be produced that slow the gastric secretions down. All right. So, and then a new set of hormones here will also increase the activity of the small intestines. Once food leaves the stomach, we don't really call it bolus anymore. We start to refer to the food as chyme. We call it chyme. As the chyme, if the chyme contains glucose and fatty acids, it causes a release of two hormones. One of those hormones is called cholecystokinins. And what this does is if there's fatty acids and glucose in the food, the stomach should probably hold on to that food longer to make sure that the digestion is continuing for longer. So the cholecystokinin, what that does is it prevents the stomach from emptying. Another hormone that is produced as food is leaving the stomach and entering into the small intestines is this hormone called secretin. What that does is that slows the activity of the stomach down. Okay, but secretin also affects the liver, pancreas, gallbladder. Those are the accessory organs. Remember that, because food doesn't enter into them. And then their combined action holds the food in the stomach for a longer period, just to make sure that that food is ready to leave. And it's ready to leave when it's sufficiently broken down. Remember, if the stomach does not agree with the food because it contains some type of toxins, well, we could vomit, right? And that's how we get rid of those pathogens quickly. This is something that we've seen before when we did the fetal pig lab. We noticed that the intestinal lining isn't flat and smooth, but it has these finger-like projections that go up and down. These finger-like projections are called villi. These are called the villi. Here we're looking at one villus. This is one villus. And what do we see? Well, making up the sides, the walls of the villus, we see these specialized cells called columnar epithelial cells. And just deep to them, we see blood vessels that make up capillary beds that enter into the body of the villus. So what's happening as nutrients are passing by, those nutrients get absorbed through the columnar epithelial cells, and those nutrients are introduced into the blood. And then the blood will leave carrying those nutrients away. It's in the mucosal layer, right, that we see these finger-like protrusions, these intestinal folds that we call the villi. Remember, the villi are important because they increase the surface area that is available for absorbing nutrients. So without the villi, the amount of area that would be available to absorb nutrients would only be this, right? Which is a smaller amount of space. But when we add villi, the nutrients could get absorbed into the blood using all of this space, right? So we have a lot more surface here that the nutrients can be absorbed through, right? You see that? This is just another image. Here, if we look at really up close, this is really zoomed in on one cell. This would be one columnar epithelial cell. This is one cell. And if you look at the edge of the cell, this is the lumen of the intestines up here. You can see that it's not, it's not a flat surface, but rather we have these hair-like projections that go out. Well, these are called microvilli. And they even further increase the surface area, right? A lot of books, you'll see this labeled with a BB. And they refer to this as, because it looks like a brush, right? So they call that a brush border. All right, so the small intestines are very, very important because, like I said in the beginning, one of the most important processes of digestion is this process called absorption, right? The whole idea, it's important to break down nutrients, right? Because when nutrients are too big, they're not usable. We need to break them down into really small nutrients so we could get them into our cells. But once they're broken down, we have to be able to get the nutrients out of the GI tract and into the blood, right? So this is the most important job, I think, of the small intestines. So it's at the duodenum, which is the very beginning of the small intestines, right next to the stomach. This is a site of chemical digestion. So digestion is continuing here. It's in the jejunum that we see absorption. So if you look at the intestines, if you take the first foot of the intestines after the stomach and cut it out, that would be the duodenum. If you take the last couple inches where the small intestines drains into the large intestines, that would be called the ileum. Everything else, which would be about, like 17 or 18 feet of small intestines, the middle part, which is really the bulk of the small intestines, we would call that the jejunum. And there's a lot of surface area inside the small intestines, about 2,800 square feet of surface area. All right, again, we see MALT continuing. We have protection against invaders in the form of Peyer's patches. They protect the lumen of the digestive tract from bacterial invasion. We see mechanical digestion happening and chemical digestion happening. Where do we see mechanical digestion? Remember, mechanical digestion is physical breakdown, right? So why would we see it with the teeth? That makes sense. We saw it in the stomach with the grinding of the muscles. That makes sense. How could we possibly have mechanical digestion in the small intestines? You would think it's just going to be hit with enzymes, right? That contributes to chemical digestion. Enzymes that break down proteins chemically, right? From the pancreas. But where would mechanical digestion be happening? I'm going to show you in a minute, and it's going to be happening with the secretions from the liver and the gallbladder in the form of bile salts. So this is just showing you the different ways to get the nutrients out of the small intestines. This is the small intestines here, this whole area. Through the small intestines and into the blood. Well, we have facilitated diffusion, so they're moving from high concentrated areas to low concentrated areas through these transport proteins. Or we have just regular diffusion, which when proteins break down into amino acids, they can diffuse from the epithelial cells of the villus into the capillaries. Here we see large globules of fat, right? So when we break these large globules of fat down, they have a tendency to want to come back together again quickly, right? Because of their hydrophobic properties. So in order to prevent that, the liver and gallbladder produce these things called bile salts. And as the fat globules are broken apart into smaller ones, the bile salts quickly surround these little globules. And what this does is it prevents these globules from finding each other and going back together, forming this other globule. So these chemicals are actually preventing the fat globules from coming back together. So for that reason, this is an example of mechanical digestion because it's a physical event that's happening. They're physically preventing them from coming back together. All right, now the enzymes like lipase can break them down more efficiently because there's more surface area here. And when they break them down, we break them down into fatty acids or monoglycerides. And we could get these to leave the lumen of the stomach into the epithelial cells by exocytosis. Remember, exocytosis, here we see active transport, getting the amino acids from the lumen of the stomach into the epithelial cells. All right, so exocytosis would require energy to do this. Active transport requires energy to do that. Diffusion does not. Facilitated diffusion does not. There's a type of bacteria called Helicobacter pylori. If this bacteria ends up in your stomach, what it can do is it can eat away at the mucus lining of the stomach. If we eat away at this mucus lining, well, what happens is the walls of the stomach become exposed and the acids can start to burn holes in the lining of the stomach. And a hole in your stomach could be something pretty painful, pretty serious. So because Helicobacter is a bacteria that would be doing this, the cure for these ulcers would be an antibiotic. Antibiotic would kill the bacteria. The mucus protective layer will be restored and that area should heal. Remember what I told you, we do have organs that play a part in digestion, even though food doesn't enter into them. That includes the liver, which is producing bile. The gallbladder stores the excess bile, and the pancreas. So if the stomach was over here, and the food was in the stomach, the food would leave the stomach and enter into the beginning of the small intestine. Remember, this beginning piece is called the duodenum, right? And it's in the duodenum that this food, or this chyme, we should call it, goes through like a car wash of enzyme activity, beginning with the bile that is going to be traveling down these bile ducts. And then it joins up with these, this is called the common duct, right? Because you can see the pancreatic ducts also join. So this would be splashing the food as it's driving by with pancreatic enzymes and then bile salts from the liver and the gallbladder. So the duodenum is a major site for chemical digestion. Chemical digestion begins in the mouth with the amylase that's in the saliva breaking down carbohydrates. It continues in the stomach with pepsin and lipase breaking down proteins and fats. But the pancreas contributes many, many different types of enzymes that breaks down pretty much everything. So this is a big site. Most of the chemical digestion of the food occurs in this little stretch of the small intestines called the duodenum. The duodenum is an exocrine gland in the sense that it produces a variety of enzymes that enter into the small intestines and chemically breaks down food. All right. In addition to that, it produces a buffer. Because remember, when the food is leaving the stomach, it's mixed with acid, and that acid could burn. So the pancreas produces an enzyme, or a buffer rather, that neutralizes the acids that is in that chyme as it enters into the small intestines. This way, those acids do not burn the lining of the small intestines. However, it's also an endocrine gland. It has endocrine function. Its endocrine function is not digestion related. It's not related to digestion. So this is an organ that is multitasking. It's preparing enzymes and secreting enzymes that break down food. So it's an important organ for breaking down food and digestion. But it's also an important organ that regulates glucose in the blood or maintains glucose homeostasis. It produces insulin and it produces glucagon. These are hormones. These are not enzymes, they're hormones. When you have high blood sugar, you produce insulin. And the insulin, what it does is it essentially takes the glucose in the blood and sends it into the liver and into the muscles, lowering glucose levels. When your glucose levels in the blood are low, it produces glucagon, the pancreas does. And what that does is that causes the liver to release the glucose back into the blood. So there's an endocrine function and there's an exocrine function. The endocrine function produces hormones that contribute to blood glucose homeostasis. The exocrine function produces digestive enzymes that chemically break down food. So two completely different jobs of the pancreas, which makes the pancreas a vital organ. The largest internal organ of the body is the liver. Now, as the chyme leaves the stomach and goes into the small intestines, the jejunum area of the small intestines is absorbing all of those nutrients. So now we have a lot of blood in the small intestines, and this blood is receiving all these nutrients. We do not want to send this nutrient-rich blood directly into circulation right away. We want to inspect this blood to make sure that it's not filled with any undesirables or toxins. So before we send this blood to the heart, where the heart can send it to all our cells, we send it to the liver first through what's called the hepatic portal system. And what this system does is it gives these specialized cells of the liver called hepatocytes access to this blood. So these hepatocytes will inspect the blood and clean the blood before this blood is sent from the intestines to the heart, removing toxins, removing anything that's in there in excess, such as nutrients or iron or vitamins or whatnot. Okay? And the liver is also at the same time monitoring glucose levels. All right. So if glucose levels are increased, the hepatocytes will remove glucose from the blood and start to put it together in these chains, right? We store glucose in the liver in these long chains, much like starch, but structurally a little bit different. These long chains are called glycogen. All right, so we take these monosaccharides called glucose and we produce long chains, polysaccharides called glycogen. So glycogen is the form that we use. So it's chains of glucose. And this is how we store sugar in the liver in that form. All right, so here we see a large central vein, and you could see that there's like these little channels, right? So the blood would fill these channels, and these individual hepatocytes are right there inspecting all the blood as it's leaving the small intestines. And then it would leave through the hepatic vein, and then it would be sent to the heart. It goes to the heart only after the liver inspects it first. The gallbladder is underneath the right lobe of the liver, and what it does is it stores bile. Bile is a product that is formed when we break down hemoglobin and cholesterol. All right, but we use it as an emulsifier. An emulsifier is like a detergent. Like I said before, if we take a large globule of fat and break it down into smaller globules, these globules will want to find each other and go back into a bigger one, but we surround them with bile salts, preventing that from happening. Okay. And that creates more surface area for our real digestive enzymes like lipase to work on those even further. Right. So this is called emulsification. Emulsification is the physical breakdown of fat globules that increases the surface area. So for that reason, because this is a physical phenomena, this is another example of mechanical digestion. All right, the large intestines has a function, and it's in the large intestines that water is reabsorbed back into the blood, but there's minerals. I tried to hit the pause button for that, but I missed it. But there's also minerals and vitamins that are leaving the food and entering into the blood as well. All right, so the reabsorption of water is the number one function. As we leave through the ileum, remember the ileum is the end of the small intestines, the chyme enters into the beginning portion of the colon or large intestines, specifically the ascending colon first, and then through the transverse colon, down the descending colon, through the sigmoid colon, this S-shaped colon, before the feces gets stored in this area, which is called the rectum. Now, because we're reabsorbing water here, if the chyme travels through here too quickly, not enough water will be taken out. And the result will be, diarrhea, right? So if there's an irritation of the colon, the chyme moves too quickly. So water isn't being absorbed. And however, the opposite problem happens when too much time is being spent inside the colon, too much water is being removed, and then the feces becomes hard, leading to constipation. Right? This is a nice table that summarizes the functions of all the digestive organs, the mouth, the tongue. Remember, the tongue is an accessory organ along with the salivary glands and the teeth. These are all accessory organs. Any organ that the food isn't traveling through is an accessory organ. The pharynx, the esophagus, the stomach, the pancreas is an accessory organ. The food never enters into the pancreas. It never enters into the liver and gallbladder. So besides the mouth, which is where most of the mechanical digestion is happening, there are other sites where the physical breakdown of food is happening. All right. The stomach is one. Remember those three directional layers of muscle that work at the same time, churning the food. So that's one. Bile, emulsifying fat globules, is another example. All right. So the mouth, in the intestines, and in the stomach. There's three areas that we see mechanical digestion. Chemical digestion is anywhere enzymes are working on the food. Remember, an enzyme is it's considered an organic catalyst. A catalyst is anything that speeds up a chemical reaction. For example, if there's a storm in the wintertime and you wake up and your driveway is filled with ice, you can get rid of that. You could speed up the melting of the ice by throwing salt on the driveway. What that does is it lowers the melting point of water. So instead of waiting for it to be 32 degrees to melt, it'll melt at a lower temperature, melt quickly. But salt is not an organic molecule. Salt comes from the ground. So that's an inorganic catalyst. Your body has organic catalysts, molecules that are made to speed up reactions. And such molecules are enzymes. Enzymes are proteins. We make them in our cells. And what they do is they speed up reactions. Without enzymes, our bodies wouldn't be able to function. So here we see a large molecule. Let's say that this is amylose. Amylose is a carbohydrate, a polysaccharide, right? Made up of these smaller units that are combined. Well, this type of carbohydrate is useless, right? We can't fit that into our cells. Our cells can't use that to make energy. So the enzyme would latch on to this piece of amylose, and a reaction would happen. In this case, this is a digestive reaction or hemolysis reaction. Of course, we need water for that, right? And we would have products. We would have smaller molecules as the products. And then this enzyme would be free to perform this reaction again and again and again. We recycle enzymes. As long as they're there, we can use them and reuse them. And the digestive system provides a large variety of enzymes. And these enzymes are coming from many different places, right? Salivary glands, the tongue, the stomach, the pancreas. You can see how many the pancreas provide, many. Even the small intestines are producing some. You could see what they work on. The substrate is what are these enzymes working on and what are they turning those substrates into? That would be the products. I remember I told you when you eat pasta, the salivary amylase in your saliva takes starch and it begins to break it down. Well, what is it breaking down into? Well, what are the products? Well, the products are going to be things like maltose. Maltose is a disaccharide, so it's a much smaller form of a carbohydrate. All right, so you don't need to memorize this chart. It's just there for your information. I'm not going to quiz you on it or anything. All right, so this concludes our lecture on the digestive system. I hope that you learned something from this. I hope it made sense. If you have any questions, don't hesitate to email me. And yeah, that's that. Okay, bye.